Assalamualaikum and a very good day to all. My name is HAYATI ZAINAL from UPM Sedang, and today I'm going to present in the MSPP 34 scientific meeting on my work entitled Salivary Protein Proteomic Identification is associated with subsequent allergic diseases in childhood. This is my presentation outline. Allergic diseases can be defined as hypersensitivity disorder of the immune system involving an IgA-mediated inflammatory event initiated in response to common environmental antigens, which include pollens, dust, and different food types. They occur when human immune system react to normally harmless antigen in the environment. Allergic diseases include asthma, eczema, allergic rhinitis, food allergies, and anaphylaxis. Allergic epidemic proportion has increased worldwide, whereby 300 million people have asthma, and they surely have negative impact on social economic welfare of the society. In Australia, a dramatic increase seen in anaphylaxis in children aged 0 to 4 years old. Hospital admission for anaphylaxis increased fourfold in the last 20 years. Food-induced anaphylaxis has doubled in the last 10 years, whereby 25% of infants now have an immediate food allergy. 19.6% of Australian population suffer from at least one allergy, and this number is expected to increase to 30% of adult population by the year 2050. Allergic diseases are the most common non-communicable diseases in children and adolescents. They are significant health burden, thus preventative measures to reduce this burden are urgently required. We believe that prenatal events play a critical role in determining disease susceptibility via environmental influences on placental function and fetal programming. Our hypothesis is that childhood susceptibility to allergy is increased through significant alteration in placental function, and the product of identified genes are altered in the saliva of allergic children. Our aim is to identify salivary protein that could be a potential biomarker to identify allergic risk in newborns and to identify target protein for early allergy intervention. In our methodology, saliva samples were collected from 9 children with allergies that comprises of 3 from children with asthma, 3 children with eczema, and 3 children with rhinitis. And we have 9 control from children without allergy. The sample were then prepared via protein extraction using 2D cleanup and the protein estimation via EZQ quantification. Later on, the protein were treated with in-solution tri-tip digestion using 5 microgram of total protein and then underwent C18 cleanup to remove contaminant that might interfere with mass spectrometry analysis. Then the sample underwent liquid chromatography, tandem mass spectrometry analysis. But prior to that, we subjected the sample to metric-assisted laser disruption ionization time of flight mass spectrometry, or also known as MALDI-TOF-MS, for quality control, whereby we try to make sure there's no polymer prior putting the sample in the Orbitrap. Finally, we analyze the result using a software called MaxQuant to quantify the protein and a mascot software to identify the protein. Moving on to result and discussion, in the quantitative analysis of salivary sample, there were 177 proteins identified to be present in salivary sample of children with allergic diseases. Out of this number, 111 proteins had more than 2 unique peptide, or UP. UP is defined as a peptide, irrespective of its length, that only exists in one protein of a proteome of interest, despite this peptide may appear more than once in the same protein. Thus, protein that had more than 2 unique peptides had a higher probability of being a G9 protein and not a false positive result. From this number, 46 proteins were identified with a significant ratio when compared to the non-allergic group in salivary sample. This figure shows salivary protein in allergic phenotypes that have LFQ ratio of more than 2 or less than 0.5 fold change as compared with non-allergic group using a max LFQ protein analysis. LFQ is label-free quantification intensity. Alright, as you can see here, the highest number of protein with more than 2-fold change were 12 in asthma group, and the highest number of protein with at least 0.5-fold chain were in the rhinitis group. This Venn diagram showing that there were three similar protein present in all of the asthma, rhinitis, and eczema group. This table shows some of the child salivary protein identification with significant LFQ ratio. This is a ratio of LFQ in children with asthma, children with eczema, and children with rhinitis. The top four protein here shows significant ratio of protein in all of the allergic children. The next two protein here shows significant ratio in asthma and eczema children. The next three show significant ratio in eczema and rhinitis children, and the remaining proteins here show significant ratio in only one type of allergic phenotype. Three protein candidates were identified associated with subsequent allergic diseases in all children, which were human mucin 5B and 5AC, human serum albumin, and human serotransferrin. Proteins in both asthma and eczema phenotype only were cysteine-S and cysteine-1, and in rhinitis and eczema only were these three proteins. The current findings suggest that protein expression varies in since utero in children who subsequently develop allergy, and these altered expression are detectable in saliva in early life. This potential salivary protein can be used as possible biomarkers of allergic risk in early life and as potential therapeutic targets in allergic disease. This is the project workflow summary. After this, we are going to choose the protein of interest, and then we are going to validate the protein with Western blot and this system, and then the final interpretation of both saliva and placental result. I would like to acknowledge these very important people that enable me to do my research. That I thank you.